life to continue, the parent must give rise to young ones which will of course carry the characteristics of the parent. But at a cellular level, what happens? The cell actually does what is called division, cell division, in order for it to produce young ones of its kind. Now, there are two kinds of division when it comes to cell division. We have the mitotic division, which is an equatorial division, which gives rise to the daughter cell that looks exactly like the parent, and the meiotic division or meiosis, which is a reductional division. A reduction division in the sense that it produces a daughter cell that has half the characteristic of the parent. So meaning that if the parent characteristic, especially we are talking about the genetic characteristic now, if the parent has 10 genetic characteristics, the daughter cell that will be produced will have only 5, which is half of 10, right? Now, what are the nitty-gritties about meiosis? Meiotic division is not a simple type of division, so we're going to be doing an integral study on the steps involved. Now, meiosis. Now, basically, meiosis is got it from the Greek word meios, which basically means to lessen or to reduce. To lessen or reduce. So that is why it is generally called a reduction division. We're going to start with something greater and end up with something smaller. That is what happens in meiotic division. Right? Now, the phases of division or meiotic division are classified into two. We have meiotic division 1, that is meiosis 1, and meiosis 2. Meiosis 1 and meiosis 2. Now, in meiosis 1, we have what is called the P-mat. P1, M1, and all of that. Then we also have in 2, we have M2, A2, and T2. Now, basically, so it means that meiosis happens in 2 stages. We have the first stage where we have the P1 mat, P-mat 1 and the P-mat 2, alright? Now, but before this meiosis takes place, there is a fundamental stage that the cell must first undergo, which is critical to the division of the cell. And that phase is called the interphase. The interphase. Now, basically, you know that it's coming from 2 Greek words that it's in between. Inter means in between 2 phases. Interphase is in between 2 phases. In between the first division that happened and then the next division that is going to happen. Alright? So what happens in this? Why is this interphase very critical for cell division to start? It is very critical because it is the one that prepares the cell for division. It is the stage in which the cell matures. With the cell, cell undergoes 3 stages. Cell undergoes division, then cell maturation or development, then cell differentiation. All right, cell differentiation. So before the cell now eventually divides, it has to go through the interphase. Now the interphase is split into 3 stages. We have the G1, S, and G2 phase. Now you see, these 3 stages are the ones that prepare the cell for division actually. Now before meiosis, before meiosis happens. So in the G1 phase, the G1 phase is an abbreviation from gap 1 stage. Then this is called the synthesis stage, and this is called the gap 2 stage. You see, in the gap 1 stage, the cell is basically maturing and growing and developing. Then in the S2 phase, the S phase, it is in the S phase that synthesis occurs, which is the synthesis of DNA, that is doubling of the DNA, such that at G2 phase we have double the content, the content of the cell would have doubled. So we have doubled the content of the cell that we started with initially. So it is at this phase that the cell matures. Here it doubles its content, and here it is ready for division to take place. Alright, now let's get into what happens in all of these phases. Now, what does P stand for? P1 stand for. Both P's stand for prophase. And in this case, it's going to be prophase 1. Now, this prophase 1 is the longest stage, is the longest phase of meiosis, of both mitosis and meiotic division. In this case, in fact, all of these 4 diagrams or 5 diagrams that I drew here represent prophase, represent the prophase stage. It is the longest stage. We have here the metaphase, and we have the anaphase, and of course we have the telophase. Now, the telophase 1, this one, alright? So now, what happens in the prophase? Basically, the prophase is split into 5 different stages, and that is why it is the longest. The anaphase is the shortest phase of the cell division. In the prophase, we have 5 more stages, which I abbreviate as let Zion play the drum. So let is going to stand for leptonema, or leptoteme stage, Basically, they are the same terms, only that this is a noun, leptonema. This is a noun, while this is an adjective. Zion standing for zygnema stage. Zygnema. Play standing for pachytene stage. Pachinema or pachytene stage. They are both the same things. Pachinema. The D standing for diplonema stage. And this standing for diakinesis stage. Alright. Now in the leptonema, this is the stage that is called the leptoteme stage. The diagram here is a stage that is called the leptoteme stage. There are 3 fundamental things that happens in the leptoteme stage. And the first is that, number 1, the chromatin, you can see scattered chromatin around the nucleus of the cell, within the nucleus of the cell. What happens is that the chromatin materials begin to coil up and become visible little bit by bit. So chromatin material begin to wind up and coil up and begin to thicken little by little. Now the second thing that you notice, that as this process of thickening and winding up starts, you are going to begin to notice what is called the chromomeric regions, the chromomere. Chromomere is just the chromomere. Chromomere is basically a part of the chromatin that is bead-like in appearance. The bead-like appearance is because of the portions of that chromatin material where we find histone proteins. Remember that the coiling up of the chromatin material is performed by histone proteins, and those histone proteins will now form junctions at certain points in the chromatin strand. So those junctions there are going to be bead-like, appear bead-like. In texture they are going to be bead-like and expanded than those are portions of more compact coiling, where the coiling happens is more compacted. So those are the portions that we call the chromomere. So those portions becomes more apparent. The next thing you are going to see in the leptoteme stage is that the homologous there's something called homology search. So what basically happens is that those chromatids, because when they coil up they become chromatids, they begin to find homologous pairs. So that is what is called homology search. They begin to find homologous pairs. Now that is what happens in the leptotin stage. And as well, the centrosome will begin to become prominent and begin to form spindle fiber. Alright? But in the sigma stage, what happens in the sigma stage or the zygote? This is called the zygote stage. In the zygnera stage, what happens is this. At this point, homologous chromosomes would have found themselves and they begin to pair up. Homologous chromosomes here would have found themselves and they would begin to pair up. And by pairing up, we say they have undergone what is called synapsis. So basically say they have synapsed. They have synapsed. So when homologous chromosomes find each other, they synapse. So when they synapse, they become homologous pairs. So you see they begin to align themselves here first. The first begin to align themselves. And by the end, it is not that it is at the beginning of this stage that they find themselves. Actually, at the beginning of zygote, what happens is that homology starch search is almost ending and they are beginning to find each other. So they begin to align themselves in such a way as they can find a centromeric region to bond. Such that at the end of this zygote stage, synapsis would have occurred. Synapsis would have occurred. So they would have joined together. Now, what happens next is we go into the next stage, which is the pachytene stage or pachinema stage or pachinema. We call it pachinema or pachytene stage. If you put stage, you don't need to put nema. So this is the now, as I said, pachinema stage. What happens at the pachinema stage is very fundamental and it's a very critical stage in prophase 1. What happens at the pachinema stage? Also notice that there are some breaking that is happening here at the cell wall. The cell walls there, the nuclear membrane, the nuclear membrane is beginning to dissolve, to disappear little by little, only little by little, especially in the pachinema. What happens here is very fundamental. What happens is what we call crossing over. And how does this result? Remember that homologous chromosomes here would have synapsed. And the synapsis has become more prominent here. So they form what is called here, but this is how synapses occur here. Chromosomes form what is called synaptonemal complex. Synaptonemal complex. Now, synaptonemal complex is simple. It's basically a complex of proteins that we call cohesin and sugosine. Now, sugosine, basically, cohesin means it's got from the word to come together, while sugosine is from a Japanese word, which means protector. Now, you see, what happens is we have a point here where these guys will attach themselves. These are homologous. This is a chromosome with homologous pairs of chromatids, right? Then you see these guys, they come together like this. So these complexes are what we call the cohesin. And everything all together is called, all of this together is what we call the synaptonemal complex, formed by the cohesin. Now, what is the sugosine? The sugosine is basically the base of all of these points here. The base of adduction of the cohesin that happens between the synapsis, the first chromatid and the second chromatid. Now, this is the reason why there is a sugosine here and it is not here. Because this portion is what forms the centromere eventually. The centromere. So, at this centromeric region, sugosine proteins are there to make sure that the coenzyme do not get attacked by an enzyme called the separase. Now, separase, the function of separase is that at the end of the Pachutine stage, what happens is that, at the end of the diplonemal stage, what happens is that the coenzyme protein becomes attacked by the separase enzyme, and those coenzymes disappear. Now, having disappeared, it is the sugar chain that is able to withstand the presence of a separase and not allow the coenzyme proteins at those regions, this centromeric region, to disappear. So those coenzymes will not disappear yet. But until the sugar chains die, when the sugar chain dies or disappears themselves or becomes dissolved, then the coenzyme proteins will also get dissolved. And that is what happens in anaphase, but we'll soon get there in a while. Now listen up. I talked about crossing over. What happens in crossing over is this. Now, those coenzymes have now formed the synaptonemal complex in between two chromosomes, homologous chromosomes. So you have a chromosome like this, and you have another chromosome like this. So you see where they are touching here, and all of this place is where the synaptonemal complex forms. Now, what happens is this. In the Pachutine stage, you are going to see that a particular chromosome, portions of each of the chromosomes at loci of each of the chromosomes, some portion of genes will be exchanged. Portion of gene, let's say this gene comes from the father, and this one comes from the mother, for example, and this one comes from the father. What happens is that the portions of this chromosome will exchange between each other, and the portions here too will also exchange genes between each other. So that at the end of the day, when they separate, they don't separate as sister chromatids, sister chromosomes again, but as non-sister chromosomes that they will not have the same genetic content as they had before, but would have mixed between the both parent traits. And that is what we call crossing over or recombination. Crossing over or recombination. Now, when they synapse together like this, they are called bivalents. Bivalents. Each one is they form what is called the bivalent. These two together is called bivalent. Now, and they now have they possess 4 arms. 1, 2, 3, 4 on both ends, and that makes them what is called a tetrad. A tetrad. Remember, tetrad for 4. So because they have 4 arms, then they are called tetrads. So this is why the first stage of the division of meiosis is called a reductional division. It's called a reductional division. Is called reductional division, while the second stage is called equational division. But I'll soon get back to this in a moment. But let's finish this first. I said that the recombination occurs where at the Pachutine stage they exchange the crossover. So now in the next phase, what is called what happens is that the separase will begin to act at what is called the diplonemal stage. So this is the diplonemal stage here. The diplonema rather, or the diplotene stage. Now in the diplonema, the sugar chain, the separase, will now attack the coenzyme, and as well the sugar chain would have disappeared as well. So those chromosomes begin to separate from each other. And the points where the recombination or crossing over of genes have occurred, which will be called the chiasmata. That's the plural. while the singular is chiasma, which are the points where the recombination has occurred. So it is only those regions that will still be in attachment. But the regions where synaptogenic complexes were formed by poison and sugar would have dissipated by the action of these enzymes separate. So when they separate, when those proteins separate, only those regions where recombination occurred will still be holding on to each other. Alright, that is at the diplonemon stage. Now, in the last stage, we have the diakinesis. In this phase, in this stage, what happens is this. We said this stage is the diakinesis stage. In the diakinesis stage, what happens is what is called terminalization. Terminalization. And what is terminalization? The points where the recombination occurred, those points which we call what? The chiasmata, will begin to move towards the end of those arms, the end of the arms of the chromosome. So they begin to, the point where the recombination occurs will begin to move towards that point. So those recombinant genes, so that those recombinant genes will be positioned on the ends of the chromosomes. So that is the term that is called terminalization. So it means that those recombinant chromosomes, we call them recombinant genes, will become terminalized. Now, what happens again is the fact that the nucleolus will already be disappearing, will have already disappeared here. The nucleolus would have disappeared, and of course the nuclear membrane would have already disappeared, and the centrioles have positioned themselves at both poles to, project their kineticals to, anchor these chromosomes. All right. So what happens basically in diakinesis is terminalization, and notice carefully that the chromosomes are still together here, only that they are held at the tail ends where the recombinant genes have moved to. So let me quickly reiterate that I said in the lactotin stage, you see that the chromosome thicken and coils. Here too, the chromosome thicken and coil, but the difference is that here the homologous chromosome would have found each other. Here homology starts, while here homologous chromosomes begin to attach to each other through a process called synapsis. So we say they have synapsed. Now after synapsing here, crossing over or recombination occurs, and after recombination has occurred, here the synaptonemal complex that is formed here where the synapse will begin to be removed here, begin to disappear here by the action of the enzyme separase, and of course only points where they are attached together, which is called the chiasma or chiasmata, are the points where they will still be remaining together. And afterwards, what is called terminalization occurs. And in terminalization, what we mean is that those recombinant genes will move to the ends of the chromosomes. All right. So with this, we have come to the end of prophase 1. So you see, there are a series of activities that happen in prophase 1. Now, what happens in metaphase? What happens in metaphase is very simple. Now, the chromosomes are going to, remember here, the centrioles are now in place and have projected their kineticals. So the non-sister chromosomes, now remember I said the non-sister chromosomes because they now have recombinant genes. The non-sister chromosomes will position themselves in a way that is geared towards being attached to the kineticals that are extending from the centrioles. Now, when they position themselves in that way, after a while they get attached at their centromeres, the centromeric regions. They get attached to the kineticals at those centromeric regions, and that process is called capturing. We say they are captured when they get attached to the spindle fiber, or the kineticals that are being extended from the centrioles. So that is what basically happens in the metaphase of metaphase 1. So the chromosomes are at the equatorial region of the cell. So they come and arrange themselves this way. They arrange themselves this way. Alright? Now, so they basically arrange themselves like that. Alright? So you see they are at the center of the cell. Now, what happens next is the anaphase. So remember, I told you that how to remember this is in the prophase is a preparatory phase. So you can say P for preparation, M for middle. So that's why they align themselves at the middle here. And A for away. What happens in the anaphase? We said that anaphase is the fastest phase, is the quickest phase of meiosis. And what happens in anaphase is that those chromosomes are with recombinant gene, non-sister chromosomes now, they now get separated. How? What happens? These places get synthesized, sensitized to move their kineticals apart. So what happens is that they draw the kinetical apart and they get to begin to move with their centromere leading towards these poles. So they get separated, basically. They get separated from each other. So this portion will get separated like this, and some of this will begin to move like this with their centromeres leading. So this is in anaphase. The centromere will be leading. The centromere will be leading because it is the centromere that is attached to the kinetical. So now, and they get drawn apart. So notice what structure we have here. Notice the structure we have here. What is getting pulled apart here is what? Is a chromosome that has two chromatids. A chromosome with what? Two chromatids. That's why it's getting separated here. Now, also notice that this is the basic reason why this is called a reductional division. Why? Because the number of centromere has been reduced to 1. The number of centromere, yes, there are 2 centromeres before. Look, when they were together here, we had 1 centromere here and another centromere. So the centromere has been reduced to 1. And what is moving here is now called a dyad. A dyad. Remember, we started at a tetrad. So what happens is that they have now started migrating apart. 2 have separated. Now they are now called dyad. Alright. So what happens next is the telophase. Now in the telophase, what basically I said think of telophase T as TA apart. So what happens? Telophase means to terminate. So it's going to then the cell invaginates and then gets split apart. The cell membrane also gets split apart, and then the cell division there is complete. Now, in humans, meiosis 2 begins after a very short, not immediately, but after a very short while. But the lag phase between when meiosis 1 starts and meiosis 2 begins is called intricacies. Intricacies. So I wouldn't be drawing kinesis. I will not be drawing any more diagrams about the meiosis 2. I'm just going to go into what happens because the phase now becomes faster here. The prophase is not as slow as before because what happens here generally, it doesn't need to thicken or wind up, the chromosomes don't need to wind up again. They only have to prepare themselves for the spindle fiber that is already forming. So the spindle fiber is already forming here, that is the kineticals. They are already forming again for the prophase. And at the metaphase 2 now, they align themselves again to the equatorial region of the spindle fiber at their centromere. They attach to these kineticals at their centromere. Then at anaphase 2, they get pulled apart. Now this is the difference. When they get pulled apart this time, it is not dyad that gets pulled apart. It is now actually monads that get pulled apart. Monads, they get pulled apart. Monads get pulled apart. So notice it is only one gene. It is only one chromatid rather. It is only one chromatid that is moving. One chromatid that will be moving away. One chromatid will be. So the sister chromatids will be separated. So notice that here there are two arms here. Two arms were separated. But here only one arm is separated. So they form what is called a monad. So notice that we start with the tetrad, then we went to a dyad and ended up at a monad. So and then what happens is they get pulled to the both ends of the pole at telophase, and then the cell invaginates and then it splits apart. So that's basically what happens in meiosis. So it is called reduction divisional because in the first place the centromeres are divided from 2 into 1 and a dyad is formed. But the second phase of division, meiosis 2, is called an equatorial division. Why? Because we end up with a monad and the centromeres do not change because we have one centromere here. So centromere is still one here and still one here. Alright, so that is why this is called, the second division is called the equatorial division. Alright, I hope you did gain value out of this video. Do well to subscribe to the channel and click the bell notification to get notified whenever I drop a new content that will be of interest to you. And God bless you. Remain focused. Peace. Or whatever.